Imagining the possibilities of genomic engineering in a massive scale. This is the topic that we propose you to explore today at BIOKET Digital. Ladies and gentlemen, good morning, good afternoon, or good evening. My name is Johan de Koning from IR, the French Bioeconomy Cluster, and I'm very happy to welcome you today at BIOKET Digital. We hope that everyone is doing fine and keeping safe. Today, we are partnering with a worldwide leading company on the field, Inscripta. Inscripta is a company that has an enzyme engineering program, and they aim to provide tools for their customers to create their own applications faster. For example, they publicly released the MAD7 nuclease. MAD7 is a royalty-free CRISPR endonuclease for both academic, commercial research, and development use. Before starting with the program of today, we have a big announcement to do. And it is not without bitterness that we announce you our decision to turn our on-site edition to digital. The actual pandemic don't let us any other rational choice. But at IR, we are resilient, and we want to turn this obligation into an opportunity. Therefore, we invite all those who could not come to France to join our digital edition. It will take place from March 1st to March 19th. All the sessions will be available on video on demand during this period. We have as well arranged a 5-day, 24 hours digital B2B. It will happen from March 15 to March 19. Therefore, there will be as well our usual exhibition with more than 40 digital booths and our startup village. 3 plenary sessions will take place on March 16, 17, and 18, and many other networking moments during that period. So stay tuned, we haven't finished yet to surprise you. Finally, before starting with our webinar, for those who doesn't know yet IR, we invite you to watch this small video.
IAR is the French bioeconomy cluster. Let's find out the 7 services we provide you to support your innovations. Tomorrow's solutions are being developed today, from ideas to endorsement and funding of your projects. Our expertise and strategic support will be there to maximize your success. Do you want to diversify your activity to reach a new market? Through our research and consultancy services, we analyze, explain, and help de-risk your business and innovation environment, enabling you to make strategic decisions and identify early adopters and clients. Bioeconomy is a European priority. Having been in Brussels since 2008, we can make sure your voice is heard. We are your ideal partner to access European funding. Innovative startup? Our Investors Club is seeking projects like yours. Thanks to our help, reach out to venture funds and raise the necessary funds to boost your project. Speed up your growth and overcome technological hurdles, thanks to the innovation platforms of our ecosystem. Our Bioeconomy Leaders Network offers you unique business opportunities. Meet your future business partners and develop your expertise at the many events we organize throughout the year. Conferences, business conventions, tech days, so many opportunities to increase your visibility and promote your activities. Grow your business abroad and give your ambitions new horizons thanks to our international missions and delegations. Brazil, USA, Canada, India, Southeast Asia, we will enhance your tech and business partnerships. Changing world, we will help you adapt your training courses to the needs of bioeconomy. Access tailor-made skills, from the endorsement of your training classes to posting jobs. IAR services are here for you. Find out more on en.iar-pol.com.
Should you like to choose our channel to promote your technology and generate leads for partnerships and commercial sales, just contact us. Nowadays, webinars are the only solution to make prospection on a massive scale. Now, I would like to invite Dr. Laura Clinton to join me. Laura is, among other tasks, the voice of the customer between Inscripta to ensure support and execution of projects related to the Onyx platform. Dr. Clinton earned her Master in Science and PhD degree at University of Copenhagen in Denmark. And before joining Inscripta, she has different commercial roles at Qiagen and Hexagon. Laura, good afternoon. It's a real pleasure for me to have you here with us today. And I would like to invite you to share your screen and to start the webinar. I will stop sharing mine first to let you the floor.
Perfect. Thank you so much. I'll just share my screen right away. So, do you see it now?
Yes, it's perfect.
Okay.
The floor is yours, Laura.
Thank you so much. Thank you, Johan. And thank you so much to IRR and BIOKET for giving me the opportunity to introduce to you the Onyx platform and how Inscripta enables genome engineering on a massive scale. So my name is Laura Clitten. I'm the scientific liaison here at Inscripta in EMEA, and I act as the link between scientists in Europe and my colleagues in the US, where you will meet two of them today. Inscripta's goal is to empower scientists whose gene editing research is stifled by current technical and licensing limitations. We will provide our unique platform and engaging in collaborative business practices, such as Johan also mentioned, we made our MAT7 proprietary CRISPR nuclease free for research and development purposes. We want to enable scientists to realize a new era of biological discovery. And Inscripta is headquartered in Boulder, Colorado, and we have offices in Pleasanton, California, and also in San Diego. And we are backed by leading investors such as Venvork, Foresight, Paladin Capital Group, and Merieux Development. Here in Europe, I will be your contact person, so please feel free to reach out to me by mail, phone, or social media if you have any questions whatsoever. And I've listed my email and contact information here so you know where to find me. And we'll also we've included some Q&A sessions for today. So please just type in your questions in the Q&A and I will try to manage that after each talk. So we'll hopefully have a good and collaborative session here. Okay, so at Inscripta, we strongly believe in the power of biology to change the world. And what we've seen over the past decade is that biology has become increasingly important in all of these different areas. I'm just going to have my pointer here. So, you know, we see engineered strains that produce antibodies to small molecules, engineered strains that produce the key ingredient of the Impossible Burger, other strains that break down plastics and other byproducts. And this trend, we believe, is just going to continue over the next decade. So really our ability to understand how biology works and use that knowledge to engineer biology to create new desirable phenotypes, we believe is going to drive advancements in all of these different sectors of the economy. And a key thing to both understanding biology and engineering biology is our ability to manipulate genomes. And in particular, our ability to manipulate genomes in high throughput. So here, there are a number of current approaches for doing high throughput genome manipulation, from classical mutagenesis, knockout libraries, or CRISPR-I or CRISPR-A libraries. But all of these approaches, while very beneficial in certain areas, they do come with some trade-offs. And those trade-offs are depicted here in the plot on the left. So here, either an approach will typically allow you to do to access a lot of locations in the genome, for example, classical mutagenesis or genome-wide knockouts, or it will allow you to make many types of edits if you're doing like protein or pathway engineering. But it's very difficult to target both a lot of genomic locations with a lot of diverse edit types in the same experiment. And there are additional challenges with these current approaches. So they suffer from a lack of precision. There are slow, laborious workflows that often require substantial automation to achieve this high throughput. It can be difficult to separate hitchhiker mutations. There can also, if you're working with a plasmid, there can also be issues with transient expression changes due to the marker or the expression system itself. So what we really want to unlock is this upper right quadrant here, to overcome these challenges and have a really reliable high throughput method for manipulating genomes to advance all these sectors of our economy in the future. And that is where Inscripta comes in with our Onyx platform. So this platform enables diverse edit types genome-wide and has a barcode-based tracking system for easy experimentation. So this is the Onyx Digital Genome Engineering Platform. It's not just an instrument, it's a whole platform that comprises of instrument, consumables, the assays to measure phenotypes in your experiments, and also the software. And this is a look of how this platform enables researchers to rapidly cycle through these experimental cycles that we call the design, generate, test, and learn, or DGTL. So starting here with design, Inscripta, the designer software enables you to design the edits that you want to make and optimize the way that these edits are going to be delivered into the genome. And after you create an order in the software, it kicks off a manufacturing step here that generates a custom set of consumables. And these are shipped to you, and you put them onto the Onyx instrument. So here are the consumables, and you have the instrument. So if we look about the input and output of the instrument, so what you do is you add your starting strain to the instrument as the input together with the consumables from Inscripta, and the output is then an engineered cell library. So we take in the instrument, we take wild type, and we also take any like your specific strain, so they can also work on this instrument. So the output of the Onyx instrument is a pool of engineered cells that we will call the engineered cell library. And let's just take a more closer look of what that is. So it's a diverse pool of edited cells. You have a single edit per cell. And in the whole library, there can be up to 6,000 edits for yeast and up to 10,000 edits for E. coli. And these edits can be diverse. They can be insertions, deletions, and substitutions, up to 63 base pairs in length. And there are also barcodes on the reagents, which are delivered to the cells on the plasmids that will enable you to easily track enrichment or depletion in experiments downstream from this pool of edited cells. So you take these cells and you can use them in a diverse range of experiments that you want to run. You can either plate them out as single colonies, and then you can put them, for example, into 96-well plates to do a direct measurement of a metabolite or small molecule, for example, using a mass spec or using a fluorescent-based readout, such a plate reader. Or you can also choose to keep the cells in a pool format and run a selection where you inoculate a flask with the pooled library that you will then subject to some conditions under some competitive growth. And then you can track enrichment and depletion of these individual designs using the barcode system. And then after you run your experiments, we have assays that help you prepare NGS samples for sequencing. And after that sequencing, you upload the raw FASTQ files into the Inscripta Resolver software, which then will analyze your data. And this allows you to link genotype to phenotype and learn from your experiment. And then this data will then be fed into the next cycle of GGTL, the next cycle of experimentation. So enable you to design new designs based on what you learned from your previous experiment. So here are some key features of the Onyx platform that I just want to go through to tell you a bit more about how it works and what it can do. So the Onyx platform works both with Saccharomyces cerevisiae and E. coli, and you can use wild type or custom strain. So the key innovation of the technology is that it uses a cell-based partition and microfluidics to enable these large-scale libraries. And with really high coverage of all of the up to 10,000 designs that you have in your library. It has an optimized editing chemistry, including our proprietary MAT7 nuclease, and the software relies on really advanced design algorithms to enable delivery of diverse edit types to the genome with high efficiency. And the resolver and designer software are just really easy to use, but they're powerful. And the instrument itself is a push button operation. So it's really easy to do those GTTLs with just 1 or 2 researchers running the entire cycle. So I'll dig a little more into the key features and how the Onyx platform works. So the really key to the platform is the Onyx chemistry. So normally when doing CRISPR experiments, researchers would have to do separately add the guide RNA to target the nuclease and then also a donor molecule to drive template-mediated repair. They will have to put those separately into individual reactions. So you could, for example, put these into 96 well plates, but that really limits your throughput. So you are limited by the plate format. So for the Onyx platform, instead we use the cells as the cells themselves as the partition. So this enables you now to have thousands of partitions in a single experiment. And the way this is done is that we have covalently linked the guide RNA targeting the nuclease, the donor template sequence, and a barcode to enable easy tracking. And I just want to highlight here that the donor sequences middle part here is what is being incorporated into the genome. The rest stays on the plasmid. But for cell-based partitions to work, we did have to develop a lot of new technology, in particular microfluidics, to keep the uniformity of the distribution of the designs in your cell population during the editing process. And that is what enables these large-scale libraries to be made with high representation of all the designs in your library. So there are also some other components to the Onyx chemistry, including the guide RNA, the donor, and the barcode. Obviously, the MAT7 nuclease to actually make the cut, and also repair proteins to help the cell's repair mechanism is also supplemented as part of the Onyx chemistry. So another key feature of the Onyx platform is our easy-to-use software. And I'll just show you one example of that, and that is the Inscripta designer. And that's shown here. And there are 2 ways that you can make a set of designs for a library that you want to do in your experiment. One is by using the graphical user interface, which is easy to learn and it's really powerful. It allows you to make substitutions, insertions, and deletions, or do a full saturation mutagenesis on either a single gene, a set of genes, or all genes in the genome with just a few clicks. And you can target specific positions inside the genes, in front of genes, and behind genes. The second way to create a library is by using the CSV file upload. This enables you to make all the same types of designs, and it's friendly with bioinformatics pipelines. So if you want to use scripting or informatically design your libraries, that's also very easy to do. So the software is really easy to use, but it's very powerful because it's driven by robust algorithms that power these high editing efficiency for all of the edit designs that you want to make in the genome. And this is driven by machine learning models that are built on very large empirical data sets and that we've collected at Inscripta. So these models have been trained on more than 300,000 designs with thousands of features evaluated in the model. And we are continuously improving these models over time. So in total at Inscripta, we've created more than 3 million designs and optimized all the aspects of a design, the guide, the donor, and the barcode that go into the cell to deliver edits that you want to make with really high efficiency. And this is one of the keys to the Onyx platform, is that you can make these diverse edit types. So you can do any edit that is 1 to 63 base pair in length, insertions, deletions, substitutions. And with these algorithms that power our design software, we are able to target more than 95% of the wild-type genome. So that means that you can target coding regions with things like triple stop substitutions to knock out genes or do a saturation mutagenesis of amino acids. And then you can also target non-coding regions to make regulatory changes in the genome, like inserting a synthetic promoter RBS in E. coli or a synthetic terminator in yeast. And here are just some examples. And there are essentially infinite ways that you can manipulate the genome with this technology. And we've just listed a bunch of ideas here, but what I really want you to think about is you could do like genome-wide truncation, saturation, mutagenesis, like we already talked about, but there are also so many other ways that you can do transcriptional or post-transcriptional regulation. So what I want you to start thinking about is what types of libraries would you design if you had access to this technology that would allow you to manipulate the genome in this scale and ultimately help you to understand how biology works and to engineer strains with phenotypes that you need. So here are just some real life examples of projects that we run internally together with a few of our early access partners. So we made it really easy to alter gene expression with insertion of a promoter ladder in front of every gene in the E. coli genome, as well as functional knockouts with triple stop codons to generate truncation of proteins. We've worked with transcription factor binding site substitutions in yeast in order to alter transcription and those expression of those genes, in collaboration with Dr. Michael Brent from Washington University. The Onyx platform makes saturation mutagenesis or single proteins or even complete pathways very easy, as you can do up to those 10,000 edits per instrument run. And we've shown with Jeff Bouquet from the New York University how our technology can be used to quickly debug strains in working on creating synthetic genomes. And finally, we've shown that the Onyx technology works on improving heterologous gene expression understanding in yeast. And these are, as I said, just some examples. So I just want to end this introductory talk with asking you to think about which experiments you would like to perform if you had access to this kind of technology. And I want you to thank you for your attention. So I want to stop sharing and introduce to you the next speaker, which is Eric, my colleague Eric Abate. And Eric, the floor is yours.
Can you hear me?
Okay, yes.
Laura? Okay, perfect. Let me share my screen here. Pointer. Okay, so now we're going to begin talking about some of the actual applications that you can potentially use for the Onyx platform. So we're going to give two examples of internal applications that we've done at Inscripta. And the title of my name is Eric Abate, and I lead the high-throughput screening group at Inscripta. The title of my talk is A New Era in Forward Engineering of Proteins, Pathways, and Genomes. Okay, so as Laura alluded to earlier, what comes out of the Onyx platform is a tube of edited cells. So it has a very large diversity of edits within that population. Each cell would contain, or all the edited cells will contain one edit within them. And they can be phenotyped in a couple different ways. So one way is you take that entire pool of edited cells and you phenotype it in a pooled fashion. So this is what's typically done in a selection. So you could take those tube of edited cells and you can put them under some sort of selective pressure. Now, certain edits within that population might confer a selective advantage under that selective pressure, and they will become enriched over time. So at the end of your experiment, you'll take that pool of cells that have been enriched in certain edits and depleted in other edits, and you submit it for sequencing using the barcoding. And then you can figure out exactly which type of edit allowed for a selective advantage and link basically the genotype to the phenotype. So that's a selection type process, which Tyson is going to talk about. I'm going to talk about the alternative workflow, where you take this pool of edited cells and you singulate them, which is typically done on an agar plate. So you plate them out on an agar plate where you get individual colonies. And then you can pick those individual colonies and phenotype them in isolation. So not in a pool setting, but each cell is now in isolation. You can grow those up and then use some sort of high throughput detection method in order to characterize them for whatever phenotype you might be looking for, such as like a high throughput mass spec technology. Once you identify the hits from your screening workflow, those then become, you submit them for sequencing. And then once again, you could link that genotype of those hits to the specific phenotype. So this is the type of workflow that I'm going to talk about. Okay, so this screening type of workflow was a forward engineering workflow where we wanted to overproduce lysine in E. coli. So lysine is actually a multi-billion-dollar a year industry, and it's predominantly used as a supplement for animal feed. And the lysine that's used in this industrial application is produced by microbes. So we wanted to see if we could engineer a wild-type E. coli strain to overproduce lysine using the Onyx platform. So in order to do this, we came up with several different metabolic engineering strategies. And one of those strategies is you can simply target an individual protein. And you can do protein engineering on an individual protein, maybe in the lysine biosynthetic pathway, to try to improve the lysine titers from that particular cell. But because you can engineer a very large number of edits at once, you can actually do protein engineering of entire pathways. So we built several libraries that target the lysine biosynthetic pathway, and we did site saturation libraries of all the genes within the pathway. And then you can also take it a step further, where you're not targeting an individual protein or several proteins in a pathway, but you can target the entire genome. So we made several genome-wide libraries. And these genome-wide libraries can target every single gene within the E. coli genome, doing, say, a genome-wide knockout library where you knock out every gene individually in E. coli, or you do a genome-wide promoter library where you put promoters of various different lengths, various different strengths in front of each gene in E. coli. And I'm going to describe that in the next slide. Okay, so this is how we designed the genome-wide knockout and promoter ladder libraries. So first of all, we wanted to make a genome-wide knockout, which we typically do by inserting a triple stop at the beginning of a gene. And you can do this in front of every gene in E. coli using the Onyx platform. And then we also wanted to make use of a series of promoters, these IgM promoters. And these are synthetic promoters that can be used in E. coli that have various different strengths. So for instance, there's one promoter here called J23112, which is a very weak promoter in E. coli. And then we picked a series of 4 other promoters of various strengths, all the way up to this very strong promoter called J23100. And what we did was, in the case of the knockout libraries, we could insert these triple stops at the beginning of every gene. So here's the E. coli genome over here on the left, and each one of these red lines shows a gene within E. coli. And we could insert this triple stop in front of every single gene in E. coli within this one library here, which had a total of 8,000 edits. Then we took each one of these individual promoters and we inserted them in front of every gene in E. coli. So here's the weakest promoter inserted in front of every single gene in E. coli. The next higher strength, all the way up to the highest strength promoter. And in total, there were 25,000 genomic edits that were designed in these various libraries. And in this way, you could very fine-tune the expression of all the genes in E. coli, all the way from a knockout, where essentially you get no expression of that gene, and then various levels of transcriptional regulation, all the way up to a very strong promoter where you'll have the highest expression of that particular protein. And then you can see what effect that has on lysine production. Okay, so what I'm showing here is the primary screening of the Onyx libraries. In total, there were 24 different libraries that were designed, whether it be genome-wide libraries or libraries that are targeting the lysine biosynthetic gene. In total, with these 24 libraries, there were 200,000 different total designs that were generated. So these different libraries were plated onto agar plates, picked by a colony picker and grown up in 96-well plates. And then we used a high-throughput mass spec phenotyping technology, which we ended up using the Agilent rapid-fire mass spec technology, which allows you to measure a lysine titer in each sample every 7 seconds. And on the left here is I'm showing the primary data. And on the y-axis, it shows the log 2-fold improvement over the wild type E. coli. And then it's showing the data from various different libraries that were generated. So you can see in the genome-wide knockout library, we saw lots of edits or lots of hits that dramatically improved lysine production. We also, as I mentioned, target the lysine biosynthetic pathway, and we used a couple different variations of site saturation libraries, which is designated by these different colors. And we could see that we got a lot of hits from those libraries as well. And then also in the genome-wide promoter ladder libraries over here of various different strengths, you can see in the different libraries, we saw hits in all of those different libraries. And in total, 18,000 isolates were screened using this particular workflow. And basically what we want to show here is that we basically saw hits in anywhere that we looked in the genome, whether it's the lysine biosynthetic pathway, a genome-wide knockout library, or the genome-wide promoter ladder libraries. Okay, so if we want to jump in and look at some of the different edits that we actually pulled out, over here on the left is some of the screening data that was generated. We actually screened these libraries in two different media. So one of them is a minimal media and one of them is a rich media. And you can see that some of these hits work better in minimal media, whereas some of them work better in rich media, and some of them work well in both of the different media types. I want to draw your attention to several of these variants were in this gene called DAP-A. And DAP-A is one of the genes in the lysine biosynthetic pathway that I'm going to talk about in the next slide in a little bit more detail. But we saw hits in lots of different genes within the lysine biosynthetic pathway. And you can see all these ones that are highlighted in purple over here were genes that we were able to identify protein variants that improved the lysine titer. We also saw a lot of hits in these so-called Y genes, or which were part of the E. coli Yome. And these genes are largely annotated, and many of them are genes of unknown function. So it's very interesting that we're able to identify variants in genes that have unknown function, but we're able to overproduce lysine. And we were able to identify 17 new targets outside of the lysine biosynthetic pathway. Okay, so I want to draw your attention to one of the genes that we subjected to protein engineering efforts, and that's this gene I mentioned called DAP-A. So DAP-A is a key enzyme in the lysine biosynthetic pathway, and it's been known in the literature, it's been shown in the literature to be a very important regulatory point in the lysine synthesis. It's known that it's feedback inhibited by lysine. So this makes sense, right? So if you're producing high quantities of lysine, or if there's high quantities of lysine out in the media, then you don't want to produce more lysine. So that the lysine actually binds to this DAP-A enzyme and feedback inhibits it. So in all the libraries that we generated, there were a total of 5,675 site saturation designs that were making various different mutants within this DAP-A enzyme. And it was known from the literature that a certain variant, which is an aspartate at position 84, when mutated to a threonine, can dramatically improve lysine titers. And indeed, we saw that in our system, and that was our positive control. So we identified numerous edits within the DAP-A gene. And in a previous life, I was a structural biologist. So one of the things you always ask as a structural biologist is, well, where do these mutations actually map to within the protein structure? So that's what I'm showing over here on the right of this slide. So this is the structure of a dimer of the DAP-A enzyme, so that the enzyme functions as a dimer. There's a lysine allosteric binding site, and I'm showing lysine bound to this lysine allosteric binding site in orange. And the position of this glutamate 84, which was known to be important for the lysine allosteric, or the ability of lysine to bind to this allosteric binding site, is shown here in blue. And what was nice when we look at the edits that we identified, which are shown in red, is that many of these edits that were identified in the DAP-A enzyme bind or are localized to this lysine allosteric binding site. So it makes complete sense, since if you make some mutation in this lysine allosteric binding site that prevents lysine from binding to it, then lysine can no longer have this feedback inhibition mechanism on the enzyme. But it was very nice to show that we were blindly screening, we would blindly design these edits and then screen these edits, but yet we're able to find all these edits clustering around this lysine allosteric binding site. We were also able to show with our screening that if you insert a very strong promoter in front of the wild type DAP-A enzyme, that you could also dramatically increase the lysine titer within that strain. Okay, this is summarizing some of the genome-wide engineering libraries that was done and some of the data that was generated. So in total, there are about 3,700 annotated genes in E. coli, and we generated 6 libraries that were genome-wide libraries. So there were 5 promoter ladder libraries, so there were 5 libraries of promoters of different strength inserted in front of each gene in E. coli. There was 1 knockout library. And with all of the screening, we saw a fold improvement between 1.4-fold to 1,000-fold improvement over the wild type E. coli strain. And we saw a bunch of novel targets, as I mentioned. So the data is shown over here. So this is showing the fold improvement over the wild type case. And we built these libraries on 2 different strains. So one of them is built on the wild type E. coli, and the other one was built on our positive control, which is this DAP E84T background, which we know already overproduces lysine. If we look at the wild type case over here, we saw that with the genome-wide promoter ladder, which is the green bars, that we saw several different edits that dramatically improved lysine titers up to over 1,000-fold in the wild type case. And then we saw a bunch of genome-wide knockout hits as well that also dramatically could improve the lysine titers. It was interesting to note that these ones that are highlighted here in blue are knockouts that target amino acid biosynthesis genes. And then in purple are genome-wide knockouts or promoter ladder that target the Y genes that I mentioned previously. It was interesting to note that when we built these libraries on top of the DAP-A E84T positive control, that we saw many more hits. And in fact, we saw very different hits than we saw in the wild type case. And you can see that many of these, once again, come from the genome-wide knockout libraries, and then also from the genome-wide promoter ladder. And this is showing the fold improvement of the DAP-A positive control by itself. And you can see that all of these show an improvement over that base strain. Okay, so I've shown you the data from the single edit strains. So then that brings up the question that, we have all these edits that target lysine biosynthetic genes that target all over the genome. What happens when we start to combine these into a single cell? So this is showing the round 1 mutagenesis, or the round 1 libraries, the edited libraries that were produced. And I'm showing you the DAP-A E84T positive control, which has about a 500-fold improvement in lysine titer of the wild type case. And then if you insert a strong promoter in front of the DAP-A gene, you get about 1,000-fold improvement over the wild type case. So then we made a second round of edited strains where we took some of these initial edits from the first round and built them on top of the DAP-A E84T strain. And what we found was that we could see quite a dramatic increase in lysine titers in many of these second round strains. But the largest edit, or the largest improvement, actually came when we combined these 2 edits, a strong promoter in front of the variant of the DAP-A E84T that no longer is feedback inhibited by lysine. And in fact, just by combining those 2 edits, had an over 10,000-fold improvement in lysine titer relative to the wild type case. So then we wanted to take and build a third round of edits. If we take some of these wild type these edits from the wild type case and now build them onto this double edited strain, what can we see from that? And that's what's shown here. And you can see in all of the cases, all of the edits that we built on top of that double edited strain, once again, can improve lysine titer. And the best strain that we have obtained has a strong promoter in front of this GLIN-D gene, which is part of the glutamine biosynthesis pathway. And that triple edited strain had a 14,000-fold improvement in lysine titer. So that's quite a dramatic improvement in lysine titer in 3 rounds of mutagenesis. And I want to draw your attention to two other things. One of them is the time commitment that was necessary in order to get this large fold improvement. So this first round of edited strains or libraries that were built took about 2 months to build and phenotype. And most of that time was spent doing the phenotype because the build can go relatively quickly. The second round of strains that were built took about 3 weeks to build and phenotype. And the third round took about 5 weeks to build and phenotype. So in the span of about 4 months, we were able to produce a lysine strain or an E. coli strain that was able to produce lysine titers at 14,000-fold over the wild type case, which is an enormous improvement in a matter of 4 months. And also I want to draw your attention to the fact that a very small number of employees or FTEs were involved in this. In fact, there was one person who was doing the building and one person doing the phenotyping. So you didn't need an army of scientists in order to build these libraries and get these dramatic improvement in lysine titers. Okay, so to summarize the forward engineering screening part of the talk for lysine overproduction in E. coli, I think we've shown that the Onyx platform allows for targeting of proteins, pathways, and the entire genome in forward engineering studies. Genome-wide libraries reveal new targets outside of the known lysine biosynthetic genes. So that allows for further areas of exploration. And that rapid rounds of forward engineering using Onyx dramatically shortens the time to engineer strains with significantly improved phenotypes, whether it be lysine titers or protein titers or any other type of phenotype you might be looking at. Okay, so that's the end of my talk. So let me stop sharing.
Thank you so much, Eric. I see there are a few questions coming into the Q&A. So Carsten Hjort is asking, in slide 12, how much manual hit inspection? Oh, sorry, that was the, apologize. How many of the designs edit did you actually get? Was one question. So
yes. Yeah, so that's a good question. We don't have the exact number of the total designs that we actually got of the 200,000. Laura, do we have an estimate of the typical number that we get? Do you, or Tyson, do you have an input for that?
Yeah, I can add a few words on that. So actually what we did when working with libraries, it's such a different way of looking at these. So we actually developed in our software, I didn't have time to talk about that today, but in the Resolver software, we actually have this, we did these algorithms that will predict how much screening do you need to do in order to actually see the number of hits. So we've these are called like the screener's curves and the selector's curves. And these are actually meant to give you some guidance when you design libraries. How do you actually make sure that you are balancing your building capabilities with the testing capabilities? So basically, we would love to explain more about that. I don't know if you can add a few words to that, Eric.
Yeah, I mean, typically when the libraries, you know, if we look at the number of the edits that we've designed that are contained within the library after they are built, typically they're greater than 95% of the total edits can be identified within those libraries.
Yeah, okay, thanks. So there's another question here from Baudouin Delphine. I apologize if I'm pronouncing names wrong here. So it says in slide 12, how much manual hit inspection did you have to do to select the candidates for the next rounds? Or did you try more combinations than what we showed on the plot?
Yeah, so typically we pared down the number of edits that we wanted to go into the next round of designs. So I'm trying to remember the exact number. I want to say it's probably around 20 or so hits that we would take from each round to the next round of library building that was done. So, but you could obviously take as many as you wanted to, but we wanted to kind of minimize the amount of diversity that we're bringing into the next round. But I think it was on the order of about 20 different edits.
Yeah, so thank you very much. I think due to the sake of time, I will stop with the Q&A, and then in the end, hopefully we'll have some more time to do more Q&A. I also want to say that if there are any questions that we did not have a chance to answer today, we will follow up and answer those by email. So you all receive answers. Just due to the sake of time, I think we need to move on. So thanks a lot for the presentation, Eric. And I would ask Tyson to the floor. So Tyson is a senior scientist in our applications development team, and he will talk about how the Onyx platform can enable genome discovery in pooled population cultivations. So Tyson, can you turn on your camera and also your microphone so we can see you and hear you? I'm not sure if Tyson is hearing me. Oh, there you are. Great.
Hey, thanks.
Okay, so thank you so much, Tyson. I hope you can introduce yourself. I apologize for being not so good moderator, but please share your screen and take it away.
Yeah, great. So I hope you can see my screen now. I should be able to. So yeah, I'm a senior scientist at Inscripta. I started approximately just under a year ago.
So Tyson, it's not in presenting mode.
Oh, thank you.
Yes.
Perfect. Okay, thanks. Yeah, so I'm a senior scientist here at Inscripta, and today I'll be telling you about one of the interesting projects of looking at it from a different perspective here, looking at it from a selectors workflow that Eric had briefly mentioned as an alternative workflow. And this has broad applications. The one being presented here is raising E. coli for the tolerance to select group of small molecules. So one of the key ways of doing this procedure currently and becoming quite a powerful technique is adaptive laboratory evolution, where you would raise your E. coli or microbe in generation after generation under continuous growth pressure in a particular small molecule that would otherwise inhibit growth. And then you're allowing natural evolution to generate mutations within the genome that allows it to become resistant to that small molecule. So this can often last for weeks long and sort of humorously, maybe even up to decades long as you grow your microbes within under particular stress. And then at the end of that, you would take that population and do next generation sequencing and be able to derive out all of the individual single nucleotide polymorphisms, but also fine insertions or deletions, discovering what's happened on the genome to allow for this, the capability of surviving in the small molecule. A downside to that is that it is time consuming. And then after you have the biochemical, after you have the individual variants that are generated, then often you need to go back and rebuild those in order to understand the biochemistry behind the mutations that were made in your pool. The other drawback to it is that it follows a fairly strict evolutionary path. So you start with a culture that's all of one genome, and then you allow it to grow out, but the growth from that is all going to operate under one mutation at a time type thing. So you don't get to explore the full possibilities of what biology could offer. So the Inscripta Onyx platform allows you to do a couple of things in this workflow that are kind of far advancing the workflow. So the first thing that the Inscripta Onyx platform enables is a high throughput method of generating those variants that you've already discovered in your AL experiments. So you can generate a library that matches all of the variants that you have previously found and get a biochemical assay going for every single one of those variants simultaneously to see which mutations are ultimately causing that tolerance to the small molecule. The other thing is that it allows for precision engineering of an initial population. So you can generate a population that's agnostic of any specific mutations and then drop that into your with your small molecule and identify new patterns that are far outside of what evolution would have normally been able to come across. So I'm going to show you examples of both of these workflows, actually. The first one is going to highlight overall how an example of how you go through this workflow. So we use the design, generate, test, and learn cycle. And here it's kind of broken out into the amount of time as well as the different workflow, the different parts of the workflow carrying through this. So you might spend a few days at the design level designing out your mutations. You would order, you get engineered cells within 3 to 4 weeks, and then a couple of days on instruments, and you would have a pool of your engineered cells that you can then take to selections. You do your selections, you take that to sequencing, and then later on I'll talk about the actual data analysis and how we can learn from that. So in this example, we worked with a faculty from DTU Biosustain, Marcus Herregard, well known within the AL fields, to be able to identify 372 different variants that were previously discovered in AL experiments. So an AL or adaptive laboratory evolution experiment was set up such that an assay was done against 12 different or 11 different chemicals, including p-coumaric acid and isobutyric acid, to identify mutations that caused tolerance to growth in these compounds. These resulted in this database of variants that are being tracked. And then that database was then converted into a library that's within that individual tube would contain all of the different variants that were discovered across all 11 different chemicals. In addition to that, we included in there 452 knockouts, which are mostly to test loss of function mutations in these genes so that we could identify because many of the AL variants that are generated are going to cause a loss of function in the gene for which the variant is. So we wanted to test that. And then additionally, one often occurrence in AL experiments is the presence of an insertion of mobile elements. And so we simulated this by doing 168 insertions of a synthetic mobile element, just to also test that capability. So to actually do this through the portal, as Laura mentioned, we have a very nice interactive designer portal. So it actually just took a few minutes to kind of recapitulate some of these AL libraries using the portal. You can see right away feedback on the designs that you've made. You can see where the designs are and how they would impact your gene of interest. So in a way, I kind of think about this when I play with it. It's a lot like being able to kind of log into a cell in a way and be able to manipulate the genome almost in real time with our algorithm, you get feedback and you can kind of see where those designs land. So it's quite a powerful software, very easy to use to move forwards into the next step, which is once you have those engineered cells, where to take them next. So we took this single tube, which contains an aggregate 992 different barcoded strains, containing each of the designs, the 372 AL variant designs, 168 insertions, and then 452 knockouts and N-terminal frameshift deletions. We took those and spread those out amongst several different growths. So we did an LB growth, a rich media growth, so that the cells would be perfectly happy. And we did M9 plus glucose as a control with no additives. And then we also did a coumaric acid additive, an isobutyric acid additive, and salt to be able to test that kind of ionic pressure. And that gives us kind of, and then we did each of those in triplicates to be able to test what the various populations containing all these mutations, how they would respond under this selective pressure. So the next part of the workflow then is we do these selections, we sequence out the barcode, which Laura and Eric have talked about. The actual barcode is the identifying factor of the design that was made. So what do we do with that data? So we want to assess the quality and reproducibility of the data across the triplicates that were generated. But key to this is we also want to identify differences in the different conditions. And then that will allow us to look for trends in enrichment and depletion across each of the compounds that we were testing against. And then we can identify new knowledge as well as compare that to existing knowledge for enrichment and depletion of certain genes. So this is a plot of the log two-fold relative abundance on the sequence counts of the barcodes. On the x-axis here is the M9 log two-fold relative abundance, and on the y-axis is the condition that's labeled here. So this is in the presence of 400 millimolar salts. There was very little difference between the growth in M9 alone versus in the presence of salts. LB, there was some growth differences, but you can start to see that the correlation is less for coumaric acid and isobutyric acid. The spread on that curve is starting to become noticeable. So this is kind of a snapshot then of the correlations. We can also then plot this to a correlation matrix, and we can see that the high correlation that was previously seen between NaCl and M9, so very high correlation. We can also see amongst the triplicates, it's extremely high correlation. So this gives us, really high confidence that what we're seeing is biologically relevant. Further, we can actually see that divergence in the coumaric acids and isobutyric acids, and even in the comparison amongst themselves between isobutyric counts and coumaric acid counts, they're pretty far divergent, and you can see that in the correlation plot as well. So it's really showing that there's distinct biological behavior that's going on between these two different growth in these two different compounds. So when you do principal component analysis, you can kind of break this down into principal dimensions. And what we see is that replicates cluster very tightly together, and also that there's a fairly large separation between the isobutyric versus the coumaric acid, for example, replicates. So again, this is indicative of there being distinct biological responses in the growth in these two different compounds that we're really able to be quite confident that we're seeing something interesting here. But of course, those are just aggregate measures. And what the barcoding actually allows is for us to dig into individual responses from every single one of those mutations that we've made. So plotting those across now on a genomic locus of the E. coli, so around the genome, is the location of where each of these mutations are. And then in the radial dimension here is the log2 treatment abundance over the control abundance. So that's going to be the coumaric acid, for example, over the M9 plus glucose control. And then also colored on the dots is the P value of it. So these are quite confident in the red dots here. And then the size of the dot is indicating the number of counts that's on that. So we can really have like very high confidence that what we're seeing is real and that it's actually, you know, counting enough that we can see that this is important, these modifications. When we compare that to the isobutyric response, we can actually start to see where some of that divergence takes place. So we can see in this region, for example, that there's some mutations which are making it less resistant or less tolerant to coumaric acid. In that same region, we're seeing some of those mutations upregulate or allowing for it to become more resistant or more tolerant to isobutyric acid. So it's a divergence in response that way. So another way to look at this same data is to do what's I think colloquially called a volcano plot, typically used with RNA-seq experiments. So this is such that on the x-axis then is plotted as to if it's less or more resistant. And then on the y-axis is the log 10 of the p-value that's shown. And so these data points that are far out here, they're more resistant because they have this higher log2 coumaric acid over M9 abundance, and also have this high p-value. And we can actually identify, because of the barcoding, we can identify the individual mutations that were made that would cause this this tolerance to appear, or this resistance to appear. Over here, you can see on the right that for isobutyric acid, it's a different or distinct set of mutations that are generated. But you might notice that RPOA, RPOB, and RPOC are showing up in many of these instances. So part of that is because these were in our designs, but then also part of that is that we're actually seeing a divergence in behavior, where we can see RPOA is landing more on the less resistance or more sensitive to isobutyric acid. And then we're seeing a divergence in response with RPOA in coumaric acid. So here I highlight the individual actual mutations of RPOA that are showing up, for example, that are being more or less tolerance in these cases. And also, like Eric, having done a little structural biology as well, I was interested in plotting out where these mutations of tolerance are, and something fascinating popped out, which is that when this is mapped onto the RNA polymerase structure, RPOA is the alpha subunit of the RNA polymerase, and they actually cluster in two distinct regions. So tolerance is generated to coumaric acid in a cluster on the N-terminal domain of the alpha subunit, whereas less tolerant or more sensitive to coumaric acid clusters on the C-terminal domain. And this is distinct from isobutyric response, which is that any of the mutations in the alpha subunit seem to have a negative impact on tolerance. This CTD of this is known to interact with transcription factor recruitments, and so it's maybe could be hypothetical that that's just disrupting the ability to recruit factors in stress, but the N-terminal domain is mainly known for assembly of the subunits. So it's less understood, I would say, as to why coumaric acid is having this clustered impact. But it does suggest that maybe there's some underlying, potentially even binding. You could imagine that what we're discovering might be a binding site in other areas, but we don't have any evidence of that. But it is suggestive that there's some very specific biological response that's going on in that region. So the other thing that we can do with with this using the same type of workflow is, what if we wanted to take that library which Eric talked about with the ladder of promoters in front of every single ORF in the E. coli, as well as a knockout for every single ORF. What if we did the same type of experiment with a growth inhibitor compound? So we can generate greater than 25,000 barcoded strains, all pooled together. And then what we did was split that apart again in LB and then in M9 glucose with no additions, but then a variety of biomass inhibitors such as vanillin and furfural that are typically generated during biomass utilization that have a negative impact on growth of E. coli. So this is now a different, instead of the 900s mutations, this is now a promoter in front of every single gene. And here we can see with doing the same type of analysis, we can see high correlation again across replicates, as well as off-axis, we can see that there's differential responses in each of the different biomass inhibitors which we used. So it really kind of shows this divergence, once again, that's present just based on the various promoters that are being engineered. So here in a similar vein, we plot this on the genomic map of E. coli. For every single gene that's present here, we have plotted all of the promoters plus the knockouts. And what's fascinating is that we can see a different response for every one of these genes that we can then make a plot. On the right, I show one example, PNTA. It's a known factor within furfural tolerance, which is now this particular compound that we're looking at. So in the presence of furfural, in front of PNTA is a medium strength promoter. In the presence of furfural, there's higher counts than compared to the control. And so then we can get a slope of that, that response. And that's what we call like our biological response in this case, for this one promoter for this one gene. But we actually have data, of course, for each of the knockouts, as well as the different promoter strengths in front of this PNTA gene, for example. So we can get the slope, that biological response for each of the knockouts, as well as each of the promoter strengths. And all these can be done in triplicate. So now we've got a slope for, as we increase that promoter strength, what the biological response is from that. What's really cool is that we can, that was just for one gene, but we have this for the 4,000 genes of E. coli. So now we're looking at all 25,000 data points that we can get from the single experiment, where we're looking at this slope, the biological response of, here's the black point says PNTA, but this is for all genes now. So we can actually see that response for every single case, allowed by that trackable barcode in the technology, the Onyx technology. So these turned out that those slope, that biological response can cluster into different regions when mapped across different biological responses over the increasing promoter strength. So PNTA falls within this, as the knockout is, it has an effect where it's depleted a little bit in the presence of furfural, but then increases as you increase the promoter strength. But others might decrease, have the knockouts be higher tolerance and then decrease with the increasing promoter strength. And so these clustering by this, we can actually pull out all of these different genes as they cluster, and then actually start to derive the functionality where these genes will be of similar function when they're clustered in regions like this. So we can really dive into new biochemistry discovery. We can recapitulate previous literature results as well, and gain new insights into, for example, how furfural tolerance can be generated within E. coli. So the creation of libraries of these cells has really never been easier. This is really like you go to this designer portal, you click through where you want those designs to be. In a few weeks, you're going to have a tube that contains all of those designs. And it's really going to open doors into new technologies to be able to do this this type of study. Yeah, and then these genome-wide libraries can then sort of push the boundaries, be used to push the boundaries for biochemical discovery. And it's also notable that in these two assays, similar to what Eric was talking about, is that because of the ease of the design, it allows for 1 or 2 FTEs to be able to generate this much data and then do that downstream analysis. That's almost like you can get a thesis worth of research out of every single time you run the instrument. So it's just spectacular to have access to this technology. Again, so this is kind of the workflow that we've went through here, where there's 2 different isolated workflow, and then that Eric talked about, and then I kind of went over a couple of pooled workflows that we can see, and really like to sort of stretch the legs on what's possible is, and Eric definitely showed this in his slides, which is that there's this ability to be able to do this combinatorial or stacked editing. And then as combinatorial edits come online, you'll be able to make multiple modifications that'll really explore outside the boundaries of what normal evolution might typically bring about. So thank you very much for listening, and I look forward to any questions as well.
So thank you so much, Tyson, for your presentation. Please use the Q&A if you have any, to the audience. We have time for a couple of questions. If there are anyone who wants to know anything about pool cultivations and the type of data analysis you can do now with this library approach. And I also just want to say that, of course, if you then realize to the audience, like, oh, I forgot to ask whatever question, please feel free to reach out to me, and I can forward any questions to my great colleagues here. So don't be shy, and just contact me if you have any questions. I don't see any burning questions coming in, so I think I'll just, thank both of you so much, and also pass it on to Johan for final. Oh, maybe something. Let me just see if there. Okay, so I will just pass it on to Johan and let you do the closing of this this webinar. So yeah, thanks a lot.
Now it's better. With the micro is better. Thank you very much, Laura, for this webinar. Eric and Tyson, that was apparently very clear because there was very little question to your presentation. It was very, very interesting to learn all the possibility that offers the Onyx platform and all the technology that you're developing. So far for the audience, for those who wants to see the replay of this webinar, let us some time to work out the recording of this webinar and to share it with you, let's say through our YouTube channel. Obviously, Inscripta Laura will get as well a copy of it, and he will be able to share it as well on the usual channel that uses Inscripta to communicate. This was a very nice opportunity for everyone to learn about the Onyx platform and all the possibility that is offering to the genomic engineering technology and to pass it this technology on a massive scale, which is, let's say, probably one of the best key enabling technology, let's say, of the last decade. It touches all the markets, not only the health, but as well the chemical market and all the biomaterials market, bio-based market, etc. It's really, let's say, an opportunity for the future to generate new functionalities, new molecules, and to enhance, let's say, the competitiveness of all, let's say, this bio-based industry. Now I would like just before, let's say, closing this webinar to share again with you the information of the day concerning BIOKET is that BIOKET is turning back, let's say, on March 2021, but unfortunately it will come back, let's say, not on on-site version, but in a digital version. We will be online from March 1st to March 19th, which will allow us to have 3 weeks on video on demand. All the program that is actually on our website will be pre-recorded on live condition, a little bit like this webinar, in fact, and it will be, let's say, posted on the platform that we are using from March 1st to March 19th with all for all the registered people. From March 15 to March 19, there will be a 5 days digital face-to-face meeting running on 24 hours, which will allow, let's say, our American friends and Asian friends to participate as well on their favorite time zone, in fact. And on March 16, 17, and 18, we opened the exhibition area. We have already 40 exhibitors that will be on digital booths. We had as well our digital startup village with more than 15 startups already registered. And then we will have 1 plenary per day, 2 hours of plenary per day during those 3 days. Obviously, there will be some networking rooms to allow people to chat, let's say, with, let's say, other participants. And we are preparing other networking moments that we will be shared with you very soon when everything will be stabilized from our side. So stay tuned with us, and because we will have probably some further surprise to announce you very next. What else to say? Probably we will ask, let's say, for Inscripta permissions, but there will be a videotheque on our platform for BIOKET Digital, and most probably the webinar of today will be there as well on video on demand. That will allow us, let's say, further possibility to watch this excellent technology and to interact with Inscripta later on if you have any question. Laura, Tyson, and Eric, thank you very much for this excellent moment of science, very high level of biotech and synthetic biology. Congratulations for this wonderful technology, and all the success to the deployment of this Onyx platform. And hopefully we get you soon back, let's say, at BIOKET Digital to propose a new technology that you are developing actually. Thank you very much. Have a nice evening for Laura, because you are in Europe, and a very nice day for Tyson and Eric. Time now for breakfast, probably for you guys, and then to work. We have finished here in Europe, but for you, it's still time to work. Thank you very much. Have a nice day, and
thank you.
See you later. Bye-bye.
Bye.
Thank you very much.